Hi everyone and welcome to another video. This video is going to be on immunity and it's going to be a very brief and simplified version, so hope you enjoy it. Okay, so before we start talking about immunity itself, we need to define it. And the definition of immunity is the process by which the body destroys or neutralizes foreign bodies. So if any type of foreign body enters the body, let's say it's dust particles or bacteria, then the body reacts in a way that gets rid of it or protects us from it. So what type of cells do we think of when we think about immunity? The first thing we have to think of is white blood cells, or also known as leukocytes. So let's get talking. Okay, so when we talk about white blood cells, we're going to classify them into two types. First type is going to be called granulocytes. The second type is going to be called agranulocytes. And it depends on whether they have granules in their cytoplasm or not. I'll start with the granulocytes. For granulocytes, we have three types. We have neutrophils, xenophils, and basophils. We're going to start with the neutrophils. And as you can see, from the word new, we can tell that it's neutral. It's granules, the granules are neutral. And when it comes to stains, neutral usually stains purple. So the stain of the granules are purple. It also has a purple nucleus with 3 lobes or 3 to 5 lobes. The function of neutrophils is phagocytosis, and we're going to be explaining what that is later on. The second is xenophils, and with the xenophils, they are acidic, and acidic substances or acidic granules stain red. So the stain here is red on the granules. So in the xenophils, the nucleus has 2 lobes, and the function of the xenophils is inflammation, and we're also going to be explaining what inflammation is later on. The third type of granulocytes are basophils, and the granules in basophils are basic, so they stain blue. Their nucleus is usually 1 lobe, but the thing about them is that the granules are so heavy or so dense or many that it covers the nucleus, so you're not able to tell if it's more than 1 lobe or if it's just 1 lobe. The function of basophils are allergies, so they secrete histamine, and we're going to be talking about that later. The next type are agranulocytes, and with agranulocytes, they don't have granules. There are 2 types of agranulocytes. We have monocytes, which are also for phagocytosis, and we have lymphocytes. With lymphocytes, we're going to be explaining the functions later on. Okay, so now we're going to be talking about macrophages. Macrophages are the same as the monocytes that we talked about a bit earlier. So they have a kidney-shaped nucleus and no granules on their cytoplasm, and it usually looks bluish. So with macrophages, what happens is that the macrophages or the monocytes stay inside the tissue in a dormant way until they are needed or until they are triggered. We have different names for them depending on where they are. For the skin, we either call them histiocytes, or they're also known as Langerhans cells. They're just known as macrophages in lymph nodes, the spleen, and the bone marrow. They're known as alveolar macrophages in the lungs. They're known as Kupffer cells in the liver. So basically that's what it comes to. Okay, so now we're going to be talking about lymphocytes specifically. With lymphocytes, we have 3 main types we're going to be talking about. The first ones are called natural killer cells. These are basically, you don't have to know much about them. just that they basically destroy bacteria, tumor cells, and transplant cells and viruses. The way they destroy these cells is by releasing granules full of perforins. Perforins basically perforate the cell membrane and kill the cell from the inside. Another thing you need to know about natural killer cells is that they kill substances which have low major histocompatibility complex. I'm going to be talking about what this MHC is. Okay, the second type we're going to be talking about are T cells. And the T cells are called T because they mature in the thymus gland, while that is in comparison to the third type, which are B cells, which mature in the bone marrow. Just note that both types are formed or synthesized in the same place. Like they are both synthesized in the bone marrow or the spleen, lymph nodes, and the thymus. But where it matures is what determines whether it's a T cell or a B cell. Now let's focus on T cells. With T cells, they have a really long lifespan. So it can reach up to 300 days. We have three types of T cells. We have helper T cells, cytotoxic T cells, and suppressor T cells. I'll start by talking about the helper T cells. With helper T cells, they are helping, so they help the functions of the cytotoxic and the suppressor. The first function is that they stimulate the growth of the other two cells. They also stimulate the growth of B cells, and they help with the differentiation of B cells into plasma cells, which we will be talking about in a bit. They also activate the macrophage system, so with the helper cells, help the macrophages get freed and attack. The cytotoxic T cells are, from the word cytotoxic, it means that it's toxic to cells. So it secretes perforins, which kill the cells or the microorganism or whatever it is. The third type is suppressor cells. Suppressor T cells have the opposite effect of the other two. It's basically that it suppresses the effect of helper cells and cytotoxic cells. This way it makes sure that the other two cells don't overreact. So it is very important in immune tolerance because it prevents the helper and cytotoxic cells from attacking the person's own body. So if you got a problem, we go to the third type, which is B cells. B cells are, just like I said, they're also synthesized in the bone marrow, spleen, thymus, or lymph nodes. It matures in the bone marrow. With the B cells, they have a very short lifespan, which can last only up to 7 days. With B cells, they're dormant. They're never actually active until an antigen is present. We're going to be explaining what an antigen is and how that works later on. They then differentiate into other things. They can differentiate into plasma cells or to memory cells. Plasma cells are responsible for secreting immunoglobulins and antibodies. Memory cells remain dormant until stimulation by the same antigen again. So that way they make the secondary response more rapid. What we mean by secondary response, it means that if you get the same disease another time. So this way the body uses the memory that's available to reduce the time of response or the time it takes for you to get this disease. That's very important when it comes to vaccinations, for example. Now that we've mentioned immunoglobulins, we're going to be talking about them a really bit. I'm going to be making it as simple as possible. We have 5 types of immunoglobulins. With all immunoglobulins all have 2 light chains and 2 heavy chains, and that is what forms one unit. IgG, it's made of 1 unit, and it has a very specific and special feature that it can cross the placenta. IgM has 5 units, so it's the heaviest of them all, and that's all you need to know about it. A has 2 units and it is passed on in secretions, so it's found in tears and breast milk as well. IgE has a function when it comes to allergies, and there's IgD. I'm not going to talk about its function. Okay, now that we've talked about the cells involved in immunity, I'm going to be talking about the actual process of immunity. Just to make this whole diagram clear. So with immunity, we have 2 types of immunity: innate immunity and acquired immunity. Innate immunity is non-specific and it's immediate, so it's the first type of immunity. It's the one that hits you first. Then we have acquired immunity. It is specific and it's delayed onset. Innate immunity is like the first line of defense of the body. Acquired immunity requires antibodies and like so on. I'll explain it in a bit. So with this whole drawing, we have a barrier. Everything on this side is innate immunity. Everything here is acquired immunity. The things in between it's a level of transition between innate immunity and acquired immunity. And then over here, we're just going to be explaining more terms that we used here. Just wait till the end. With innate immunity, as I said, it's the first line of defense. So the first type of innate immunity is natural barriers. So they are natural things found in the body that help prevent the foreign bodies from entering. First one is skin. Obviously, it's just a barrier. We have gastric acid in the stomach. It usually destroys bacteria and so on. We have tears. We have mechanical reflexes like coughing, sneezing, vomiting, and tearing up. And then we have commensal or normal flora. These are basically bacteria in the intestines or in the rest of the body, and they manage to compete with other bacteria. So if these are available and they're good bacteria, there isn't enough nutrients or enough supply for other bacteria to survive. Then we're going to be talking about the second response, which is inflammation. Inflammation is a complex of tissue changes as a response to injury. It promotes wound healing. So the mechanism of inflammation is that there is vasodilation of blood vessels, increased capillary permeability, clotting, migration of cells, usually of white blood cells, and this is known as chemotaxis. We also have release of histamine, prostaglandins, cytokines, and activation of the complement system, which we will be talking about later. So also prostaglandins are basically local hormones. Cytokines, we're going to be talking about them later. There's also clinical signs that are visible with inflammation. There is swelling, and that is due to increasing of the capillary permeability. It allows the fluid inside the capillaries to migrate or to move to the tissue, so it swells. We have redness and hotness, and those two are due to the vasodilation of local blood vessels, and blood vessels look red, and also blood vessels emit heat. We have pain, and that is due to a lot of factors. I'm not going to be talking about that, and there's also loss of function due to the fact that the organ or the tissue is injured or damaged. Then we have other mechanisms. We have natural killer cells, which you've already talked about earlier. We said that they destroy the cells by perforance. We have the complement system, which we will be talking about later. And we have phagocytosis. Phagocytosis is basically the ingestion of bacteria or foreign body by endocytosis. Endocytosis is the process is known as cell eating. It's the process of engulfing a substance in a vesicle and entering it in the body. After that happens, the vesicle gets fused with digestive enzymes which digest and destroy the bacteria or foreign body completely, and that way we got rid of the foreign body. Okay, so up until the point where we digest the bacteria by phagocytosis, we call this innate immunity. Then we go into the part which is between innate and acquired. This is called antigen presentation. Antigen presentation is basically that after this phagocytosis happens, let's talk about bacteria, for example. Bacteria has certain proteins or certain parts that trigger or are recognized by the body, and those we're going to be calling them antigens. Okay, the antigen is so when the bacteria is phagocytosed or it's ingested or endocytosed into the cell, so when the bacteria is taken into the cell by phagocytosis and digested, it will keep this antigen or this polypeptide or protein that's an antigen to use it. Okay, so this protein that's digested is complexed or added to this other substance known as MHC, which I talked about earlier. It's called the major histocompatibility complex. Then this mix of antigen and major histocompatibility complex is presented on the cell surface of these. There are specific cells which their main function is to be antigen presenting cells, or also known as APCs. Antigen presenting cells are macrophages and B lymphocytes. They have the ability to present this antigen on their surface. Other cells also have the ability to present antigens, basically all nucleated cells. So with the major histocompatibility complex, we've got two types. We have type 1, which is always found in all nucleated cells. Type 2, which is only found in antigen presenting cells, and T lymphocytes or T cells. T cells, lymphocytes only respond to MHC antigens. So what do we mean? As we said, innate immunity is all of those. Acquired immunity is immunity that is achieved by only lymphocytes, and T lymphocytes only respond to MHC antigens. So that means this step is vital for lymphocytes to attack. Okay? So let's go to the acquired immunity. With acquired immunity, we said it was specific, so it only destroys a specific thing, and it has a delayed onset, so it only happens after the innate immunity happens. We have two types. We have humoral and cell-mediated. With humoral, it's when it is related to antibodies in circulation, or in other words, it's related to B cells. We have cell-mediated, which is about T cells, activation and the function of T cells. So the first thing that happens is that we activate the T cells. The T cells are activated how? When they see MHC antigens, okay? So we activate the T cells. We already explained the functions earlier about how the helper ones stimulate the other ones, and then how they activate or secrete perforins, which kill the microorganism, and they also stimulate the B cells. So then, step number 8, which is that the T cells activate the B cells. The B cells then form plasma cells, which form antibodies and release them in the blood. And then the 9th is that we form a memory. So the next time those memory cells see this foreign body, they recognize it immediately and trigger the whole immunity process faster. Okay, so now we talk about the complement system. I've brought this up mentioned it a few times here so far, so let's focus on it. The complement system is basically made up of 20 plasma proteins. They're named as, the ones that we're going to be more focused on are C1 to 9 and B and D. So we've got like Those ones are the important ones. There's a lot more, but these are the ones that matter the most. But I'm only going to be talking about the C's. I'm not going to be focusing on the details of each pathway. I will just be talking about what triggers it and the main common pathways between them. So, the first pathway is known as the classical pathway or the classic pathway. This is a pathway that is triggered by antigen-antibody complex. So, when the antigen and the antibody meet, or when the antibody sticks to the antigen trying to destroy it, we trigger it. The second part is the alternative pathway. This gets triggered by bacterial endotoxins. So the moment a bacteria enters the body, it starts secreting endotoxins, and these endotoxins trigger the alternative pathway. The third pathway is the lectin pathway, and basically there is this lectin that binds to mannose, or mannose is a sugar that can be found in those foreign bodies, and it binds to it, and yeah, the process is triggered. So what happens is that we stimulate or activate one of the complements. Whenever you activate a complement, it changes from the number to an A and a B. So for example, if we have a C3, we change it to a C3A and C3B. Okay, let's talk about how, what happens is that we stimulate one, which stimulates another, up until they form an enzyme known as C3 convertase. Just to note that C3 convertase is not the exact enzyme in each of the pathways. So there is a C3 convertase here, there is one here, there's one here. So after that, we form this enzyme called the C3 convertase. The C3 convertase then converts the C3, which is the inactive form, into C3A and C3B. C3A and C3B. C3B then binds with the C3 convertase to form a C5 convertase. The C5 convertase converts the C5 into C5A and C5B. Then the C5B joins with C6, C7, C8, and C9 to form C5B 6, 7, 8, 9. And this is basically known as MAC. MAC is an abbreviation for the membrane attack complex. Membrane attack complex function is that it attacks membranes, or it destroys it, therefore destroying the foreign body. There's other functions of complement systems we're going to be talking about. We have C3B, which is known for opsonization. Opsonization is that if it sticks to when this complement sticks to a bacteria or a foreign body, it makes the bacteria more susceptible to phagocytosis, or it makes it tasty. So it makes those phagocytic cells digested or ingested. The C5A is for chemotaxis, and we already mentioned it earlier, which are agents that attract white blood cells. We have C3A, C4A, and C5A, which help the release of histamine from mast cells. We have MAC, which is responsible for cell lysis. This is pretty much it when it comes to complement systems and immunity. I'm going to be quickly going through it again. Let's imagine that a foreign body enters. It has to go first through the natural barriers. So if it's ingested, it has to go through the gastric acid. It can be attacked. It can be protected from with the skin, or if you like breathe it in, you can sneeze or cough. If you ate it, you can vomit. Those are other ways. If it's bacteria in the intestines or somewhere else, the other good bacteria can compete with it and prevent it from working. Then we have inflammation, which is if this foreign body damages a cell, the cell is going to go through all of these events. These events help with, first of all, if it's an injury, it'll help clot and close the injury. If it's not an injury, it'll help attract more white blood cells to the area, and it'll also release other substances which help, such as histamines, prostaglandins, and cytokines are hormone-like substances or molecules that act to regulate the immune response. So like they can be secreted by those white blood cells to help regulate the immunity. They can activate cells. They can also help with the differentiation of the B cells. They can do chemotaxis as well. And obviously, their main function is promotion of inflammation. What happens with inflammation? We've got the natural killer cells, which can kill it. We can also activate the complement system, which will release MAC, as we said, and MAC can cell lysis. If that doesn't happen, we've got the phagocytosis. We take it into the body, and we can destroy it. After it's destroyed, we form antigens, which are basically the polypeptide of an antigen with MHC. This can stimulate the T lymphocytes. The T lymphocytes then try to kill the organisms again. Then we activate the B cells, which activate the plasma cells, which form antibodies. Antibodies do the same function. They go around the blood. Now, here's the thing about antibodies. It's good that they're not localized, so they can go through the blood. They can kill all this foreign body in the body. And then we have memory, which we save it just in case this whole thing happens again. That is pretty much it, and thank you so much for watching.